Hey guys, so today I'll be talking about the synthesis as well as the secretion of thyroid hormones. So basically, there are 7 steps in the synthesis and secretion of thyroid hormones. So the first one is trapping, oxidation, iodination, conjugation, endocytosis, proteolysis as well as release. There is also another step that is in between trapping and oxidation where the iodine and ion is actually getting released into the follicular lumen, but it is just not mentioned here. So basically there is like 8 steps, but here we are just talking about the 7 main one. So now we are going to talk about the we are going to get into the details of all these 7 headings. Okay, so this is the rough picture. I can't get any better picture to explain the synthesis and secretion of the thyroid hormones. So yeah, this is the overall picture of all the 8 steps that I have mentioned above. So the first one is, as you can see here, just now we talked about the first step is the trapping. So this is the first step that is happening here, trapping of iodide and ion. Okay, the synthesis of thyroid hormone begins with the trapping of iodine by thyroid gland. The iodine and ion, this one I minus, is rapidly absorbed by the gastrointestinal tract and is actively taken up by this transporter is also known as a Na plus I minus symporter, or also known as a NIS, located at the basolateral membrane of the thyroid follicular cell. So this is actually the thyroid follicular cell, and this is actually the basolateral membrane. So apart from this I minus anion, there are also several other anion that can actually compete with this I minus anion. Those are actually perchlorate, pertechnate, cyanate. Okay, those are the 3 anions that actually can compete with this I minus. So those 3 anions actually can be used as a medication to, let's say you're actually having hypothyroidism. So those 3 anions can be used as a medication to reduce your hyperthyroidism, because all those 3 things will actually prevent this I minus anion from getting inside. So when there is a competition for this I minus anion to get inside, so the synthesis of the thyroid hormone, synthesis as well as the secretion of the thyroid hormone also will get lesser. Okay, then the iodide which enters here will then leave the follicular cell and enter the follicular lumen across the apical membrane. Okay, and this transporter is also known as a Cl minus I minus exchanger, or also known as a pendrin. Okay, Cl minus will go inside, I minus will go outside. So Cl minus and Na plus will eventually combine. Okay, so this transporter, if it is mutated, it can cause goiter as well as hearing loss. Okay, those are the first step that I was talking about, which is actually about the trapping. The second step is actually about oxidation. Okay, in parallel with the secretion of I minus into the follicular lumen, the follicular cells also will release this thyroglobulin into the lumen. Okay, this thyroglobulin contains a tyrosyl group. Okay, they actually contains tyrosyl group, to which this I minus will actually attach. But I minus cannot attach directly. They must be converted to the atom form of iodine. So there is an enzyme called thyroid peroxidase. This thyroid peroxidase will actually oxidize this I minus to I atom in the follicular lumen, and this reaction requires water, which is provided by another apical membrane protein, which is called as dual oxidase 2, which is not visualized here, but there is apical membrane protein which is called a dual oxidase 2, also abbreviated as duo X2. Okay, so that is actually the second process. Okay, first, second, oxidation. So after you oxidize it, you will actually get the iodine atom. So, one or two oxidized iodine atoms incorporate selectively into specific thyrosyl groups of the thyroglobulin. Okay, now we are actually talking about the third process, which is also known as the iodination. So, this process where the iodine atom incorporated into the thyrosyl group of the thyroglobulin, this one is actually catalyzed once again by the thyroid peroxidase, and this process is also known as the iodination or organification. And the end product of this is it will generate single or double iodinated species of the tyrosine, termed as a monoiodotyrosine, known as a MIT, okay, MIT, or diiodotyrosine, known as a DIT. Okay, so that is all about the third process. Now we are going to talk about the fourth process. Okay, the fourth process is actually how we are actually producing the T3 hormone or T4 hormone. The thyroid peroxidase, once again, the third function of it is they combine MIT and DIT residues to generate T4 or T3 species within the thyroglobulin protein, and this process is known as coupling or conjugation. T4 is generated by combining two DIT, whereas T3 is actually formed by combining MIT and DIT. So you know, MIT means 1, DIT means 2. 2 DIT means it's 2 times 2 equals to 4. That means it is a T4 hormone. MIT is 1, DIT is 2. 1 plus 2 is actually equals to 3. That's why we call it as T3 hormone. Okay, one important thing that you have to remember here is TPO enzyme or thyroid peroxidase enzyme is more efficient in combining 2 DIT than to produce T4 than combining 1 MIT and DIT to produce T3, which explains why thyroid gland primarily produce T4 more than T3. Okay, later, the peroxidase process thyroglobulin is then endocytosed by the follicular epithelial cells on a regulated basis whenever the thyroid gland is stimulated to release hormone into the circulation. Okay, this process is also known as endocytosis. So, importantly, peroxidase processed thyroglobulin within follicle can act as reservoir for thyroid hormone in the absence of stimulation of hormone release. Okay, even though let's say your thyroid has been shut down, you still have some sort of stores here that actually can be released into the circulation in order to compensate for the shutdown of the thyroid gland. Okay, so that is all about the endocytosis process. Now I'm going to talk about the second last process, which is also known as the proteolysis. So once endocytosized, the TBG combines with the lysosome. So as you can see here, it's not well stated here, but the TBG or the thyroglobulin will actually combines with the lysosome, and these lysosomal enzymes then degrade the TBG into T4, T3, DIT and MIT. Okay, T4, T3, DIT as well as MIT. Okay, this MIT and DIT, they will be catalyzed by the enzyme called deiodinase in order to produce I minus and anion once again, and this I minus will actually go inside. The another function of the deiodinase is actually to convert the T4 to T3, because T3 is the active form of the thyroid hormone, T4 is the inactive form, T3 is the active form. So in order to release the thyroid hormone, T4 would actually be converted into T3. It's not as if all T4 is actually converted to T3. Some of the T4 will also be released into the circulation just in the form of T4, but some of them will be actually converted into T3 by the enzyme deiodinase. So this is basically the two functions of the enzyme deiodinase. The first one is to convert MIT and DIT to I minus anion once again. Another one is to convert T4 and T4 to T3 and release them into circulation. Okay, both T3 and T4 is released into the circulation by the facilitated diffusion, and MIT and DIT is then salvaged and then transported back into the follicular lumen as I minus anion. So one thing that I want to mention, like this question is very popular in exam. usually the single best answer questions. So they can even ask in the short answer paper questions. So what they will be asking about is, what are the functions of the thyroid peroxidase enzyme? So you just have to know that the thyroid peroxidase enzyme actually has three functions. The first one is oxidation, converts the iodide and ion into iodine atom. The second process is iodination, where the iodine atom is incorporated into the thyroxyl group of the um, thyroglobulin. And the third process is actually the thyroid peroxidase is actually catalyzing the addition. I wouldn't say addition, it's like the incorporation of the MIT and DIT, or DIT and DIT, to produce T4 and or T3 hormones. So those are the three important functions of the thyroid peroxidase enzyme. So that's all for this video about the synthesis and secretion of thyroid hormones. I'll catch you guys up in another video. In the next video that I'm going to talk about is about how the thyroid hormone is actually going to act on the specific nucleus receptors. Okay, thank you guys.